Thanks, Corey. And thank you to everyone visiting in person as well as out in Zoom land. This is so exciting for me to be at the Draper Museum. I've gone to presentations over the years, and I really feel honored because I entered this very stellar cast of people who have presented to you. And I've been really honored to work with Corey. He's always been so gracious over the past years. When I text him or call him or email him, and at the last moment, can you give a tour to my insert class here? And he's always gracious, squeezes my students in at a moment's notice, and changes their lives. And ultimately, that's what I'd like to do today, is perhaps plant a couple of seeds in your minds, in your hearts, and show how I hope that I've been able to do that for a host of students through the years. Actually, one of them, well, I see one present student here, and I see one former student, Stellar Chloe Winkler is back there, and you'll see Chloe when she was a little bit. Oh, and there's Tara. Hi, Tara. Awesome. And JJ is over there. Thanks for coming. So, what are my goals ultimately in studying diseases on the landscape of the Greater Yellowstone Ecosystem? Certainly scientifically, I want to characterize this landscape, and birds experience a significant number of diseases. So that's goal one. But I have a probably equally important goal, and that is to provide opportunities for undergrads so that they can learn about research, not as an aside, but as an integrated, important part of their science, technology, engineering, and math education. I was really lucky. I had phenomenal professors when I was at Montana State University in Bozeman initially, and then at Oregon State to finish my bachelor's, that really opened the doors to me. And I want to do that for these students as well. And I couldn't do that without saying thanks. This is undoubtedly a list that I've missed people on, and I'm not going to read them all, but this is a multi-person endeavor, and one of the persons is right there in the third row, and that's Dr. Alan Childs, who has been a partner in this crime for some time, and notably in the second row is my wife Melanie, up top and underlined, because I couldn't do this without Mel's passion, without her support, without her understanding when she has to wait for me while we're hiking because I'm identifying or e-birding a bird in particular. And I also want to mention that this project is funded by the IDEA Network's INBRE program. INBRE is a program that is funded by the National Institutes of Health, specifically for biomedical research. Biomedical? Yeah. In Wyoming, this counts. Birds carry disease, and what we do is we share these opportunities for others to learn. And I happen to use birds as a model. So with that in mind, let's talk about birds. Thank you, Corey, that I've never had the opportunity to talk about birds for 4 hours like you've given me today. No. I'll keep it short. But birds are like the stars. They really are. They're accessible. They're accessible to everyone. We hear them. We see them. We've noticed this for millennia. Our cultures, there are at least in the Greek perspective, there are at least 8 constellations that are avian. Shakespeare mentioned at least 65 types. Think about it. Birds awaken us with matin, and they lullaby us with vespers. They create our worlds, societies, and cultures. Birds bring news. They announce birth. They announce death. Birds feed us, trick us, scold us. Birds awaken our dreams, and birds give us hope. The Shoshone people know Heart Mountain as home of the birds. This is on my commute every day from Powell back home to a farm outside of Belfry, and I snapped this out the window of my Yaris the other day, just because it highlights a couple of things, I think. The sun is setting over Heart Mountain. Heart Mountain is known as the home of birds, and I think that's just fascinating. It's the gateway. It's the gateway to Yellowstone. It's a gateway to the GYE, the Greater Yellowstone Ecosystem, and these harbingers of good and bad news are birds so often. And I didn't worry about those power lines in there because they were vacant in that photograph, and it kind of is prescient, because some days it seems like that hope has flown away. This is a study that came out in 2019. Since 1970, North America has lost approximately 3 billion birds. 3 billion. That's 3/8 of our population in the world. This is substantial. I cannot run into people. I can't not run into people that bring this up. They don't see the birds they used to see. They miss the birds they used to see, because many of those birds were the birds that always accompanied us to work, accompanied us to school, on hikes, or the birds that watch us watch them out in the yard. So this is something that we notice as humans. And this is a question that kind of keeps me awake at night. What will we do without them? They still fly in my dreams, and they always have since I was a little kid, as Corey alluded to. And they still sing in the recesses of my mind all the time. So why ultimately do I study birds? I want to introduce others to these avian wonders. I want to meet new friends, both human and bird. Notably, they're both bipeds. I want to meet these new friends who, even now with declines, we can still find them. I want to understand the world, and ultimately, because I must. I was called to study birds. That's why I study birds like this young American robin. But birds are suffering, and among many other factors, there are many factors that are being identified as causes for the loss of our avifauna. Is disease a significant stressor here in the Greater Yellowstone ecosystem? So overlaid upon this robin, I have just a few diseases that these birds are working through. West Nile virus, for example, the viremia that they experience, and also are they able to overcome it with antibody, with immune response? Going clockwise, malaria. Birds do suffer malaria, just like people suffer malaria. It's different species, so no, you cannot catch the same malarial protist from mosquitoes as the birds do, but there are at least 3 genera, Plasmodium, Hemoproteus, and Leucocytozoan. We're assaying those. Coccidia also, that's an intracellular intestinal parasite. Some of our birds are hit with those, and interestingly, those are species specific. A Wilson's warbler will have a different coccidial species than will a yellow-rumped warbler. Bacterial community, they carry bacteria as well. So I look at the gastrointestinal or fecal microbiome, and one bacterium that has been a significant driver in some population declines is Mycoplasma, and that's conjunctivitis that you've probably heard of or maybe even seen. And then, of course, there are other viruses ever waiting in the wings. Avapox virus is one, and you probably read the newspaper article or the note the other day, avian flu, highly pathogenic avian flu identified in Canada geese south of Laurel. So this is really complex. These are all working among the birds. There are over 330 species of birds in the Greater Yellowstone. I concentrate on a subset, the passerines, the perching birds, the songbirds. And thus far, I've assayed nearly 60 species. And just as each bird and bird species is not unto itself, each disease is not unto itself. So this is really complex. Are there diseases that are working synergistically or antagonistically? These are questions that are just now being asked in human health. Kind of this 1-2 punch. How about asking it in the avian world? And meanwhile, everything is changing. Climate is changing and it's becoming much, much more significantly variable. So that's kind of what sets the stage. So in science, we ultimately base everything on models, because models are not correct, they're not truth, but they help us to hang what we know and on them, like on these bars, and then also help us to identify questions. These are two different models, and you may have heard about the one on the left as COVID started. That's an SIR model. That is applied for microparasites like viruses, where you have those people or those birds that are susceptible. Then they get attacked, they become infected, and then they're removed from the population through death, or they recover. So that's one of the models that I use with respect to West Nile virus and bacterial diseases. And then there's another model, which is macroparasitic, which is more complex in ways, and these are those larger parasites, those larger pathogens, where you have a host population of birds, and then these pathogens have to mature, and then they go out onto the landscape, sometimes through a vector. And you'll notice that vectors can play a role in both of these models, linking through that American robin. In the middle, up on top, you might have West Nile virus being carried by Culex tarsalis. That's the little mosquito that has a white band on its proboscis, so you can identify those just by sight. In the summer, malarias are carried by some other mosquitoes like Aedes or Anopheles. Anopheles are the mosquitoes that are head downers. They'll land on you and then they'll put their head down like this. And then there are also other biting dipterans, no-see-ums, biting midges. They're carrying some other of these pathogens. So this is really complex. Really complex. And my models are probably laughable when we really look at the complexity of this system. But this complexity really breeds the opportunity to learn little bits of it and try to pull some of those questioning arrows into focus. So ultimately, I involve students. We involve students. Alan and I involve students. And we have a new addition, Austin. I just saw Austin back there, who will be bringing people in. Through disease ecology research, I introduce college students to the wonders of birds. Thus far, specifically, 35 students have directly helped me with avian research. Over 20 additional students have participated. We've had 28 undergraduate posters or presentations, 5 at professional meetings. These are professional parasitology meetings or ornithological meetings where my students who are 18, 19 years of age, community college students are there with graduate students, and they get these phenomenal experiences. We've heretofore assayed over 975 individual birds of 25 families, 6 orders, sampled for this study since 2013, and our number of in-brace students just keeps rising. Students are introduced to all these different aspects. Literature review. What is science? Compliance and ethics. That's an important part. We're federally funded, so we have to go through compliance and ethics training. How to sample vertebrates, data collection, blood collection, etc., all the way through. How to develop a curriculum vitae so that they can move on. How to go to a meeting and not sit in the corner and be engaged. And ultimately, genomics, bioinformatics, and what I believe, passion for birds is passion for science. They take that with them, and some overarching concepts that they learn. Diseases are dynamic. This year might be completely different than last year. Host populations are dynamic. Vectors are dynamic. Coevolution is occurring. The landscape is dynamic. Change, change, change everywhere. Weather and climate. Models can help us. Practicing science ultimately is about relationships that are dynamic. Birds are cool, and ecology ultimately is a circle where everything is related. So how do we do this? We go out on the landscape and we catch birds. We use mist nets and tube feeders, potter's traps across an altitudinal and habitat gradient. We have each bird in hand, we age and sex it, we take morphology measurements, what we call morphometrics. We put a numbered band on, so when you're driving down the highway, you see a dead bird along the road, please stop safely, check it out to see if it has a band on it, and then you can report that band. And many of you know this as well. And if anybody's a duck hunter, you certainly know this. But we learn a lot of information, even yet, by just affixing these bands. And each bird is processed and released within 20 minutes. Like this just stellar Audubon's warbler, this male yellow-rumped warbler. Taking an oral swab, here we're gathering a little bit of blood. We take a whole bunch of processing data, and I'll go through these here in just a second. Mist netting. This is really a skill. You have to be permitted. You have to demonstrate your ability to do this, and it's under federal and state permitting that I may not have mentioned, but that was on my title slide or second to the title slide. All of this is permitted under regulatory statute. And I train these people to do these young people. This is Kayla and Chloe in Dr. John Campbell's yard. John Campbell contributed many of these photographs. Thanks, John. We take morphometrics, the stellar red-winged blackbird male, looking at his bill. It has been recognized for ages. Actually, you can go to Darwin's second edition of The Voyage of the Beagle, 1844, and he alluded to this. He was too shy to really talk about evolution by means of natural selection yet, but he was talking about the bills of Galapagos finches and mockingbirds that he was most interested in. But we measure bills, and bills are their hands and their mouths when you think about it. So ultimately, they're really under strong selection to have a well-adapted bill, and that's a really important aspect that we measure, among others. We take saliva, we take about 1 microliter sample on a little swab, and this will give us an estimate of viremia, the number of West Nile virus particles that are in its saliva. Data gathering, I love this photograph. Everybody is so busy. We have birds in hands. Each one of those bags has a bird in it. And sometimes we just have to shut the net because we have so many birds, we cannot process them because we're trying to get them through in a low stress situation so that then they can be released. We bleed them. This is common grackles. They are such a raucous, crabby, depredating bird. They always remind me of the Nazguls from Tolkien's Lord of the Rings, but they're just beautiful in this almost wicked wicked way. And there, this guy is nicely giving us some really bright blood. We take about 70 microliters is all, one capillary tube. We photograph their morphology. For instance, the eyes of the grackles are really fascinating. We band them. We affix bands. You might notice there's a couple of colored bands. On house sparrows and house finches and American tree sparrows, we color band them so that we can get a reciting. And I have birds at our feeder at home outside of Belfry, the house sparrows that are 5 years old now. And they're still out there wearing their little color bands and coming to the feeder and raising their nestlings every summer. And we let them go. That is a significant part of it, is always to be kind and be ethical in our use of these little creatures. My students also gain really valuable laboratory skills. Both these young women got an opportunity to go to San Francisco last February with me to learn side by side with a very gracious disease biologist named Ravinder Sehgal in his lab. This is Carlos helping us out. And my students really recognized the importance of laboratory skills, got some great experiences, and really understood the importance of teaching others to learn those skills too. Some of the skills that we fight with a lot, and I was fighting with this yesterday, are bioinformatics and genomics. We actually try to teach students coding at the command line, and this is a significant change from using your phone. And ultimately, we're looking at gene sequences, or even lower than that, nucleotide-based sequences, trying to understand the types of bacteria that are infecting or living commensally with these birds. And we make things called taxa bar plots. That is that kind of wild graph that happens to be northern shrike data there. So ultimately, I take this approach. Students respond to their interests, all the while through parsing and analyzing subsets of data. They contribute their own parts to the whole. That's what we try to enhance in the INBRE program. So what I'm going to show you coming up are a lot of student-derived perspectives and student-driven questions that over the years these young people have endeavored. So what are we learning ultimately from the birds, including this male house sparrow? A little background first. We all know that West Nile virus has been around for a while. It was characterized in Uganda in 1937, I think. It didn't really arrive, as far as we know, until 1999 in New York, and since then has spread throughout the nation. And this is a disease that is vectored by Culex mosquitoes, and is one that you and I can get. And it can be fatal to humans. It can be fatal to birds. It can be fatal to horses. Usually not to dogs or to cattle. It is everywhere. It is everywhere in the United States, except for a few counties, and it has really rapidly moved west with a vengeance through the prairie potholes, Fremont County in Wyoming, Riverton, that area. That's just a hotbed of West Nile virus. And think about it, irrigating, you have a lot of water on the landscape, warm nights, all these things going on to increase West Nile incidence. And there's a seasonal pattern. I want you to try to memorize this graph. This is in humans. Notice pretty clean, relatively speaking, through the winter. There's a little uptick in May, June, July, August. Mosquitoes on the landscape, people are getting ill, then it kind of tapers off again. That's humans, so memorize that. Let's look at our West Nile virus prevalence. This is a proportion of the birds that we've encountered by month. or by year. 2013, it bumped up in 14, down in 15, back up in 17. If I was not careful, I would say, oh, obviously a 4-year cycle. No. To show cyclicity, you have to have a much longer series than that. But I wouldn't say that it's a 4-year cycle, even though these kind of look that way. I think any cyclicity we would need dozens of years to really show. But it ebbs and flows. Okay. Remember the other graph. This is birds. Kind of up there in the upper right, there's a compiled graph in human. Look at this. January, none. February, there's an uptick. March, how many mosquitoes are you encountering in a usual March? And Culex tarsalis, no. If you do, it's usually in 80s. What is going on here? Why do we have these birds early in the year that are showing high levels of viremia in West Nile virus numbers? Fascinating to me. This is something, And then it kind of goes down, and then up in June. I would kind of expect it in June, and then kind of tapers off through the fall. I have some hypotheses. The birds are starting to go into breeding behavior. Their hormones are rallying. Their physiology is changing. Are there West Nile virus particles latent in their bodies that then get released when they undergo stress of breeding? I don't know. Who has it? Across the board, these are the big ticket items. Across the board, with regard to prevalence, these are the birds that experience West Nile virus, and these are alpha codes, which is the way that many of us in ornithology think. And I'll go through them. Wilson's, from left to right. Wilson's warbler, Bullock's oriole, house wren, red-winged blackbird, gray catbird, American robin, house finch, common grackle, house sparrow, European starling, American tree sparrow, brown-headed cowbird, American goldfinch, western wood pewee, and the yellow warbler. And you'll notice that there are a couple of those species that are relatively high, over 33%, Wilson's warblers, over 20%, Bullock's orioles, and then it just kind of tapers off. So everybody and their canary seems to have West Nile virus. I left off a couple of species. I left off sage thrasher and green-tailed towhee because I only have caught 2, and both of those were positive. And it's kind of interesting to think about where sage thrashers are. They're out in the sagebrush. Green-tailed towhees are up on Hart Mountain and all, and how are these guys getting West Nile virus? And also, Wilson's warblers are ultimately birds that breed generally higher than Culex tarsalis, the mosquito, lives. So where are they picking this up? Lots more questions all the time. Another student she wanted to really investigate these families of birds and try to assess not just do they have West Nile virus or do they not, but how much of a load, a viral load. And actually, this is a logarithmic scale here over on the left. It's called a ramp value. It's a type of ELISA, which is an assay that uses light to detect the virus particles. And you can see the family that is really notably there in the middle. The red line going across indicates kind of a positive count, is the Mimidae, the gray catbirds. Gray catbirds can just be loaded with West Nile virus particles. House sparrows can be loaded as well. Yeah, Corey told me about this. There we go. Here can be loaded, but not really like this, but certainly here are some outliers. Those are house finches, largely. And they're another species that can just be loaded with West Nile virus. And interestingly, many of these species in the literature have been shown to have an association with West Nile virus where it's 100% fatal. We're finding house sparrows recovered. We're finding yellow warblers recovered. Yellow warblers were known to recover, but house sparrows, it was assumed to be fatal to them, and they recover. Shrikes. Shrikes are related to the corvids, the crows and ravens. And crows and ravens are very sensitive, we have historically thought, to West Nile virus. And it has been assumed to be 100% fatal in those allied birds like vireos, shrikes, and all the crows. And we've had shrikes, northern shrikes in particular, that have cleared the virus as well. So coevolution, are they able to develop an immune response? And I really think that we're seeing that in our black-billed magpies, maybe, but magpies are really hard to catch. They're so smart. And I don't have a magpie sample. So that's on the docket, as well as another corvid, as Corey mentioned, pinion jays that we'll be looking at forthcoming. But no bird is unto itself. This is a first jab at some network analysis. This is going to be, I think, so fun analytically over the next year or two that I'll be working on this. But we're going to try to model these interactions between these birds that are positive for West Nile virus at the same location and same time, and develop a model to where we expect that there are going to be big drivers, like maybe the catbirds, maybe robins, house finches, maybe, that are kind of spreaders, potentially. But again, West Nile virus does not, knock on wood, usually, transfer laterally without going through a vector. And the vector is usually Culex tarsalis, but some midges do it too. Other students have endeavored to look at condition index and try to graph out birds in poor condition. Are they more highly loaded with West Nile virus? Maybe, but it depends upon their size. Over to the right, that's a better condition. Over to the left, that's less of a body condition is what I'm talking about. And then the West Nile virus going up higher here. And it kind of depends upon the band size of the bird. So you're kind of quantifying this or qualifying this in bins of how large this bird is. And maybe those like house sparrows and house finches, those all were in that band size 1, 1A, 1B, 1s. They may be showing a pattern that's different from other sized birds. Elevation. I think Chloe actually was working on some elevation. You can see prevalence there in the mid altitudes, but once we get above 5,000 feet, we're not seeing West Nile virus, and that is corroborated by the literature. Higher you go, less West Nile virus. Higher you go, less malaria, also generally speaking. How about the landscape character? This is, again, West Nile prevalence. Over there on the left, again, prevalence is the proportion of your population or birds that you catch that have West Nile virus. There's a lot of variability in the rural landscape, the wild, the ex-urban interface, whereas in urban sites, the prevalence is higher and there's not much wiggle room there as well. We have two notable house species here that are not related. House finches are native, and house sparrows are non-native introduced species, but they cohabitate. They're with each other all the time. They're at the feeders together. Are they experiencing something different? Yeah, they are, to where they're showing a little bit difference in the viral loads. A little bit. There's a lot of overlap, though. So we can't really run to the bank, I don't think, with this yet. How about malaria? There's a little bit of difference. House finches on the left, house sparrows on the right. Looking at blood smears, we take blood smears from these birds as well. And there's a difference, 39% to 32%. These are kind of ballparks, okay? There's a little bit of difference, as one might expect. And how about their immune response? You can assay kind of a sense of how these birds are responding immunologically by counting white blood cells, and then comparing that to how many red blood cells you've gotten, et cetera, kind of trying to standardize it. And house finches really seem to be on guard. They have a lot of white blood cells. Why? I'm thinking they might be hit by a lot of pathogens chronically. So they're ramping up their white blood cell production and activity. But I want to take a little bit of a detour right now. I alluded to this before. We're working through pipeline called a bioinformatics pipeline, where we assay all of these different types of bacteria that are living with, I don't want to say infecting, but living with these birds. So as a subset, I took a subset of birds from our population, and these were all captured by my colleague Abby Valine, who has done phenomenal field work. And we assayed northern shrikes, and I had to put in northern shrikes. This is my favorite species. This is what I got my master's on. And I duly love this interesting and wicked and beautiful singer, northern shrikes. And you can see in the bacteria that we assessed, almost 90% are in this one phylum, this group, proteobacteria. Then we get three others that contribute not much to the diversity. So what the heck is proteobacteria? These are species like, but I'm not saying that shrikes have them. Brucella, which you may have heard of, Escherichia, Haemophilus, Vibrio, Pseudomonas, Bordetella, those are common genera. Firmicutes, Bacillus, Listeria, Staphylococcus, Enterococcus, Streptococcus, those gram-positive bacteria, if you know a little bit of that. Actinobacteriota, Streptomyces, these are generally soil microbes. And then I think this one's interesting, Desulfobacteriota, which is only 0.2%, but these are sulfate-reducing bacteria. And I wonder if shrikes have them because they're very carnivorous, and that might be associated with protein breakdown. That's just a hypothesis. But what ultimately did we find? Really no significant pathogens. Pseudomonas can be, but it's a very ubiquitous and abundant genus throughout the world. And it really rules as far as what was found in the northern shrike droppings. Yersinia, you might know Yersinia pestis, plague, Black Death, sylvatic plague. There are several species of Yersinia, so we could not tell if this was indeed Yersinia pestis. Haemophilus influenzae, you might know about this. It was found in a female. It's really everywhere in Coxiella, that is a tick-borne bacteria. So that's kind of interesting, but no Borrelia, which causes Lyme disease, no Vibrio that causes cholera, among other things, no chlamydia. So ultimately, nothing notably pathogenic, bacteria-wise, were the northern shrikes carrying. Here, so how do we kind of go forward? Are there some questions that we want to investigate? Yeah. Host density, fomite. Fomite is like a non-living object, like your phone, that can then, you can pick up and catch a disease from someone's phone. That's a fomite. There's a lot of concern about feeders. I really think in the vast majority of cases, bird feeders, especially the non-platform, are not functioning as a fomite, and data have borne this out, maybe with conjunctivitis. the house finch eye disease, Mycoplasma gallisepticum. But other than that, probably not. Urban lighting is really interesting. It seems to be lengthening the infectious period of West Nile virus, so yard lights, street lights. Warm nights over 17 degrees C seems to increase West Nile virus capacity and also malaria, and also those mosquitoes being able to breed. So look at our warm nights that we've been encountering over the past few years. And ultimately, our house finches being a driver in this system, but look at this poor little guy over here on the right. He has conjunctivitis. And he had quite a high bacterial load. He had a West Nile virus load. Is he just getting hammered? And are they just becoming exhausted through immune system exhaustion? And I wonder if this is really going on with some of our passerines. And others are more robust, and they just get through it, kind of like house sparrows. So going forward, I want to further foster student initiative, exploration, determine recovered population. This is really tough. West Nile virus antibodies are tough to elucidate because the assays have been made for chickens. And chickens give a lot of blood. And they give more blood than my little bird weighs. So you can't use those same assays. So Dr. Jason Gigley, who is a toxoplasmosis researcher at University of Wyoming, has generated a new ELISA, a new procedure for assaying toxo. And he thinks that we can tweak it for West Nile virus. So I might be able to do with just a 1 microliter of serum and assay if this bird actually is mounting an immune response. Because if a bird is mounting an immune response, that's good news unless it gets out of control. So this is what we're looking forward to in the future. Parasite cascade, develop some migration models. Obviously that grackle, those 2 grackles, that's a female on the left, that's her iris. Fascinating features. That's a male on the right. The males exhibit a really clean demarcation there, where the females are often kind of blurred out. And what's going on there? But they migrate. Are they picking these pathogens up on their wintering grounds? Lots to learn. So finally, we have to end on hope. We live in the age of birds. I know everybody says it's the age of mammals. We're in the Anthropocene, but birds outnumber us inordinately, just on species number and individuals. So my charge to you is to learn about them. Share your world with them because they share their world with us. There was a screenshot of my eBird the other day. eBird is like Pokemon Go for biologists. It can be very addictive to note every single bird. And I know John and I are religious about getting at least 1 checklist in a day. And oh, heaven forbid if we miss a day because we're going to lose our streak. But this is an introduction to people who don't know birds too. It gives them the ability to contribute to citizen science, like Corey brought up, and Sue. Hi, Sue. I haven't seen you in ages since you worked for the Park Service. Talked about the Christmas bird count. Get involved in Christmas bird count. Midwinter bald eagle surveys. Share it with other people, and it will do you good. Right there. Colleagues at VPI, Virginia Tech, are studying how humans respond to bird feeding, how humans respond to bird. It makes us better people. Ultimately, people are satisfied, are happy. Project Feeder Watch, all these citizen sciences, but even if you don't do that, just share your world with birds. This is a poem. I'll see if I can get through it without having tears. This is a poem written by our daughter, Callie. My wife, Melanie, is sitting in the audience, as I mentioned earlier. This is Callie. Callie is a poet. She's a teacher at Joliet High School, teaches English. September 22nd morning. Without knowledge of the past, we shall lose the future. Of cottonwood leaves, drinking in sunrise, becoming gold around dark-eyed juncos, silhouettes. Reflections on water caught in the sunken yard from a thunderstorm that brought down the dead maple at the end of the road, leaving us dark-eyed and candlelit. How long can we live in darkness? Seeking equilibrium, I balance, calling on strength in my core. Where do juncos find their strength? Do they call fleaish to balance their world? Should I? No matter how long the day, the evening comes, dark-eyed, along this road. And my parting message to you is a yellow warbler that we named Gamgee, who bred at least 3 years in our yard, and he was West Nile virus positive, and he cleared it and came back every year for a multiple number of years, probably from Costa Rica, where he spent the rest of his time. See if this works. Be strong, be free, may the force be with you.
It's like, why are you touching my screen? There we go. Don't go into the net.
Not into the net. Yeah, we don't want him to go into the net. So thank you for your attention. I appreciate it.
You'd be willing to repeat questions and then the microphone for our
Definitely.
So, first question I have from an online audience member is pertaining